start from there. This is that part of the chapter which we are already doing, and the chapter name is basically called as protein synthesis. Okay. And for protein synthesis, we have already done different types of ribosomes. We have already done, the functions of these ribosomes and what is the difference between a ribosome and a DNA. Okay, we have already done. But now we are going to discuss that, this is the phosphate group, which gives the nucleic acid their acid in nature. That's very important because sometimes examiner asks from you that why we used to say the term called as DNA, means deoxyribonucleic acid. Why you is using this term over here as an acid? What thing makes DNA acidic in nature? Okay, so that thing we need to remember, that why you are talking about it, that it's a DNA, or you are saying that even it's a ribonucleic acid. Why are you using the term of acid over here? Which thing is making it acidic? So you need to remember one thing, as you know that you have this, DNA with one ribose sugar, as you have already, this is a pentose, and the phosphate group is attached over here. And then obviously there are different types of nucleotides depending upon its guanine, cytosine, or uracil, or thymine if it's RNA. So this is the phosphate group which is going to make it a bit acidic, and we'll discuss in detail that it makes the acidic in nature, that DNA, and that's why we used to take this name as acid. Okay, then we are going to discuss another important thing that's called as phosphodiester bone. Okay, it means that there will be two ester bonds. Okay, let's have a look now. So here, this is, let's suppose, you can see this is phosphate group. Okay, this is a pentose sugar, and the R group, which is going to be, different for different amino acids. You can check this amino acid have different, this amino acid have a different, and this amino, there are different kinds of amino acids. You can say that they are having different kind of, R group attached to it. If I'm talking about nucleotides, then you can find that, okay, there are two types of nucleotides. One is called as purine, and the other one is called as pyrimidine. Okay. So you need to remember these two, that pyrimidines will be the single ring sugars like this. And purine will be the double ring structure of the sugars like this. So you can see the double ring over there. Anyways, here look at one important thing. You need to see that two hydroxyl group where on the phosphate molecule are going to bind on 3 and 5 carbon on two independent pentose sugar. Okay, have a look now. As you know, the numbering of carbon will start exactly just beneath this the oxygen group over here. So this is 1, this is 2, 3, 4, and this is 5. This is pentose sugar. So look at the carbon number 3. I'm talking about here, and then I'm talking about carbon number 5 of the other. Okay, now look at the other one. So starting from here down, 1, 2, 3, 4, and 5. You can see that this is your 5th carbon. Okay, now what you need to check? You need to check that the pentose sugar having 3 carbon attached to the phosphate group, and look at here the 5 carbon of the other, this pentose sugar. So there are two independent pentose sugar, number 1 and number 2. So what you are making in between them, you will be finding that there will be 2 hydroxyl groups over here, which you will be using to make a bond over here. These are 2 condensation reactions taking place over here. So at the end, you will find that 2 water molecules will be produced from here. So that will be the production of 2 water molecules. You can even check over here as well. If the same thing you are following, then can you see that this is carbon number 3 and this is carbon number 5 again, and you can find then again the bond is between this carbon number 5 and carbon number 3. So what is happening that the phosphate is bonded to the sugar. Look at here, this phosphate is bonded. Let's suppose here this phosphate is bonded over here. Then it means that if it is bonded by the bonds, we say that there are 2 bonds which are helping them to bond over here. One bond was formed here and the other bond was formed basically here. So what we term them, we term that there are 2 ester bonds present over here. That's why we use the term phosphodi, means 2 ester bonds. And which reaction, this condensation reaction is catalyzed by exactly two enzymes. And there is an important enzyme which we use the term called as ligases. I used to say that if it's a G over here, think that like a glue. So this ligase always acts as a gluing effect, mean to join something. And this is a very important enzyme when we were discussing DNA replication. We will come to know that what DNA ligase do. And another, I hope you remember from the plasmid, we did from genetic engineering, that was the insulin production. I hope you remember, we cut the part of the plasmid and then we inserted here the human insulin. And I told you that these are the enzymes. We don't have the scissor to do these simple works that are very minute details. So what are these things, or you can say the substances which are used to cut and then to join even at the micro level? So they are the enzymes. So for cutting we have restriction enzyme, for joining we have ligase. So that's why the term is used here, the ligases. I hope you remember now. You will not forget that there will be two ester bonds between the 3 to 5 linkage. What you need to remember from here, 3 carbon to 5 carbon linkage between the two pentose or ribose sugars. And similarly, it will continue to the next because we are talking about a polynucleotide. So how polynucleotide is formed? That there are a lot of peptide bonds. Right now, we just discussed about the phosphodiester bonds. Okay, now basically the DNA molecules are made up of two polynucleotide strands, right? And how they are standing one in this direction and the other one will be obviously the anti-parallel direction, opposite to direction. We have already discussed that if let's suppose it start from 5' to 3', then the other will be from 3' to 5'. So it will be exactly opposite to one another. Why you need to do this anti-parallel, and why you need, because you need these two strands to held together by some bonds. And you can see that, as I already told you, the mnemonic between G to C, that is government college. So you need to remember that G to C, there will be always 1, 2, 3 hydrogen bonds. But adenine will always pair with thymine, it will always have 2 hydrogen bonds. So for this bonding, you need to remember that you have one strand of polynucleotide in anti-parallel direction. Look at here. This thing will be clear to you more. Look at it. This is 5' end and this is 3' end. You know that adenine is always going to pair up with the thymine. So if you remember, if I'm saying that okay, let them into the same direction, what will happen? Can you check? Can they be pairing up with one another? So just imagine 5 over here. What is over here? This is cytosine, but we need thymine to be over here. So the pairing will not be happening. So what we have to, this adaptation is that if this strand is running from 3' to 5', the other will be run in opposite direction. So now in this way, they can have these bonds between them. I hope now this thing is more clear to you people, that how these anti-parallel strands to one another are going to make these hydrogen bonds with one another. Okay, have a look again. So DNA molecule have one strand anti-parallel. So the two strands are running like a helix. They will be running like this. They will be like helix. Okay, and you already know that the backbone is basically a pentose sugar and a phosphate group. And I told you that why DNA is acidic in nature, because it's having phosphate in its backbone. We have already done this, but quick revision that what are purine and what are pyrimidines. So purines are basically the sugars which are called as adenine and guanine. Okay, so they are double ring sugar. While pyrimidines are single ring sugar, you can check it's thymine, and if it's uracil, then it's going to be in RNA and then cytosine. Okay, have a look on it. Have a look on the sugar structure, having no oxygen over here at carbon number 2, but yes, having oxygen over here. So deoxy means not having oxygen at carbon number 2 over here. So I'm quickly just repeating again the two rings, which are purines and pyrimidines. You don't need to remember the structure, but you need to remember that purines are double ringed, and obviously purines are guanine and adenine. Okay, while for pyrimidine, you need to remember it's single structure of a ring, and then it's the examples of the pyrimidines. Got it? So this is the way you need to remember the, and you don't need to remember much about the bondings in between. But sometimes some questions for P1 comes when you need to recognize them by the double sugar and the single sugar. Okay, here you can see again, and I told you that one complete turn, if it starts from here, it will have how many base pairs? 10 base pairs. Okay. So, and we have already done this pairing up. I'm just quickly swiping this. You know that this is a double helix, you know, the base pairing. But today we are going to start a very important topic of DNA replication. Look at the two important scientists. One was called as James D. Watson, and the other is called as Francis Crick. So they had a very important experiment, and they performed experiment on DNA, and they basically found out that it's the double strandedness of the DNA. They say that genetic molecule must have been copied many times. And these two scientists, Watson and Crick, suggested that there are two strands of DNA, and they can even split apart as well. So they can basically unwind first, and then they can even split apart. So basically, according to them, replication, that's very separate from the cell division. You know, we have already done the cell cycle, mitotic cell cycle, and we have already done that there is a phase which is called as interphase, and then comes the mitotic cycle thing. That is the PMAT. And at the end, you started that it's a cytokinesis. That's the end of the cell division or cytoplasmic separation. So interphase is very important. I hope you remember you started that there are three stages in it: G1, S, and G2. Okay, now you remember we did in S that there is synthesis of or duplication of the DNA. So that's what we have to study here today. So what happens that appropriate partner term was first of used by them to form the complementary strand. Okay. So the new DNA means that in interphase, what is happening? We have, let's suppose this DNA, then we have to make a copy of this DNA again, and this new DNA would be just like the old one because each base would only pair up with its cap. Like look at this. If this is having same guanine to cytosine, then obviously here will be the pairing up will be the same one. And what will happen? We can even bind up again into a double helix exactly like the original. So it can be, binded again. So that's an important question regarding your replication of DNA. That's called as semi-conservative. Okay. So that's very important question of even P2, and sometimes it comes in P1 as well. So this is an important question. I'm marking star on it so that now be attentive what we are going to learn today. So during replication, we know that there is a double-stranded DNA. We are using the term molecule. What it happens, it first of all, step one, separates. Like this, if it's a double helix structure, then it's going to first of all separate or unwind like this. Then this one is going to act as a template for the synthesis of new DNA over here. And that will be called as a lagging strand. So we'll discuss that in later, but right now you need to remember that it means that DNA molecule, which is a double-stranded, must have first unwindingness, and then this unwindingness will basically create a newly synthesized another copy that will be called as this one. This one will be called as a newly synthesized DNA strand. So we can say the old one and the new one. But this is called as semi-conservative replication. So what is this? Look at here. I'm talking about the unzipping first. Can you see? That the first step is DNA replication is the unzipping. And it's not done by, very easily. You need some enzymes. You need enzymes for the unzipping. And the enzyme which is responsible for unzipping is called as helicase. Means you remember A's from that ends with A is called as enzyme. And if it's going to unwind the helix, then we term it as helicase. What it do? It basically caused the two strands of the DNA to separate. Look at here. So what it has basically broken down over here. What were over here? They were basically hydrogen bonds. So this enzyme basically broke the hydrogen bonds between these nucleotides which are present over here. So what it did? It basically, it's kind of a enzyme which is going to exactly split the DNA into two halves apart. So DNA double helix is going to unwind or unzip, and the halogen bonds between the bases will be breaking. So this is the first step of the DNA replication. Okay, what will happen then? There are free nucleotides present all around the nucleus, and they have even extra phosphates. Got it? Even you can see that these base pairs are added with extra phosphate which are already freely present over there. So what helicase do? Helicase basically completes the separation of the two strands, and meanwhile the free nucleotides, they have been activated by the attachment of these phosphates. They start the addition of these two phosphate molecules on the complementary strand which is going to be formed new. Let me just explain it again. What is the first step? Number one, the helicase. What it's going to do, it's going to unzip it, unwind it. Step number two, what is happening? That the phosphate, which is already present over there in the nucleus, they are going to attach with the free sugar bases which are already present, like adenine, thymine, guanine. The moment phosphate will add, they become active. We use the term active. Now these nucleotides are very much active to be attached on the complementary strand. Let me color the complementary strand with the blue color. So here this is the complementary strand. These active nucleotides can come, and if this is having G, which nucleotide should come over here? Obviously, that will be the cytosine to come over here. If it's T, then which nucleotide will come here? That will be adenine. The pairing will be like that. The pairing will be the complementary pairing, exactly which is the rule of it. Okay. Now look at here. This thing, it's more clearer to you. So each of the bases which are now called as activated nucleotides. Why they are called as activated nucleotides? Because the phosphates, they were added. But the moment now they will attach to this, now these phosphate will even released. So what is happening, that activated nucleotides will pair up with the complementary. Let me color the pairing of the complementary with another color. Okay. So you come to know that this is the pairing. This one is the pairing of the new thing. This one is the pairing of the new thing, complementary. Can you see? We have marked them as a complementary strand. This is the original strand. This is the original strand. It was, unwinded by an enzyme called as helicase. There was an enzyme that was called as helicase, which unwinded it. And then there were free nucleotides present over there, which activated the nucleotides over there by the addition of the phosphate. Now, there is another enzyme that's very important. The name of that enzyme is called as DNA polymerase. Repeat with me, DNA polymerase. So what is the function of DNA polymerase? The function of DNA polymerase is basically linking the sugar and innermost phosphate groups of the next door nucleotides together. Okay, can you imagine now? The linking of all these active nucleotides on this complementary strand, like here and like here. And who is doing this? The name of that enzyme is called as DNA polymerase. So the two extra phosphates, when they will, obviously sugar will be joined, they will be broken apart. They will be released again back to the nucleus. Got it? You can see that they are releasing now back to the nucleus again. What we need over here? But we need one phosphate. Why? Because the nucleotide is basically this in the structure. Let me draw it again from over here. So this is a pentose. If I'm talking about a pentose, and then this is a phosphate. You need a one phosphate over here, which is going to be exactly over here, the backbone of the nucleotide. But the rest of the extra phosphate, they will be released. Okay. Now have a look on the DNA polymerase. What was the function of the DNA polymerase? To add on the complementary nucleotides in front of the, or the one which was winded. So the new nucleotide is going to be having all the incoming nucleotides bonded by the enzyme called as DNA polymerase. We can term it as a growing chain. Okay? The growing chain. There might be few mistakes even while, if there's some binding, there might be few mistakes, and perhaps it's around after every 10 is to power the 8 bases. There might be. So in your course, it's not much in detail, but let me show you something over there. Okay. I hope you remember the structure. First start from here. So if there is a one strand of DNA, the complementary strand was antiparallel. And this is the starting of this topic. Okay, so this is the antiparallel strand, which is running over here. Now, what important thing you need to remember, that DNA replication can be initiated at many points, but always from 5' to 3' in direction. From 5' to 3' in direction. That's you need to remember. Or if I'm talking about here, from 5' to 3' here. It cannot initiate from 3'. That's the important thing you need to remember. Okay, so it's happening in from 5' to 3' direction of the replication. What this part is called as 1 and 2. We term it as sometimes replication fork. The forks where replication starts. So step 1, what happened? The helicase was the enzyme which basically unbinded this double helix. Now it's uncoiled for the clarity. So it's unwind by the helicase. But where? Only in the regions where there will be some activity going on for the DNA replication, and that region is called as replication fork. Like here, like here. Okay, these two things are clear to you? Then we can move forward. Number 1, replication always starts from 5' to 3' direction. Number 2, to unwind we have helicase. To basically attach the new nucleotides on the complementary strand, we have DNA polymerase. You need only to remember this. Now come on this diagram. We told you that DNA polymerase is helping you in basically assembling of the complementary strand. And you can see that it started from prime to 3'. What is helicase doing? Helicase is basically, look at here, this is the helicase. After that, you can see now the DNA strand is unwinded, uncoiled. And these are the pool of nucleotides which are freely present. I hope you remember the term ligase. Ligase is always used for the sealing up the new fragments, the new nucleotides which are born. Got it? Now, tell me, there is an enzyme which is called as DNA polymerase 3. You can see that even we have 2 and 1 as well, but in your syllabus, it's not in much detail. Okay, so this is the point from where we know that DNA replication will start. Helicase is only going to have one function, that is to uncoil. What is the function of the DNA polymerase? That is to assemble these, freely nucleotides on the complementary base pair. And what this ligase is going to help you out? To attach, to paste, to basically seal those fragments. Got it? So this is the way how your DNA will start its replication. And here you can see DNA polymerase is adding nucleotides. So in all, let's revise again the function of DNA polymerase. This is the enzyme which traveled down the DNA strand. And what is the only function? The sole function is to add the nucleotide bases to each strand. And what about ligase? I told you for G, you need to remember glue. So ligase seals up the fragments into continuous strand. So sometimes even this question also comes for the difference. Now we were discussing about the DNA replication possibilities. We discussed that it is semi-conservative, but before that, we have to go through a few possibilities. Let's suppose if we are thinking that it's conservative replication, in which you will find that there will be whole one new double helix and one will be having its daughter. Like if this is the parent, the old one, this one, then these are the two daughters, 1 and 2. This will be the exact replica of the parent. You can see the color. We are telling you this will be the exact replica. And this will be the daughter nucleotide, you can say the DNA. So we can say that the new complementary double helix will be made from the old one directly whole. That is one possibility. This is called as conservative. Everything is conserved over there. But what semi-conservative? That's very important thing to keep. Have a look. Can you see the red color? Let's think that it's a daughter cell, daughter strand, or you can say daughter strand of the DNA molecule, and this one is from parent. So this is very acceptable, can say that so it was an experiment which was done by even straw, but we will be discussing it later. That semi-conservative replicated, it is accepted one as well. That whenever we have a newly synthesized DNA, it will have one older strand and one new strand synthesized. So what happens? It says the same thing which we have already done. That number one, the DNA molecules split into two separate strands. Okay, that done by the helicase. And then what will happen? The replicated one will be the mirror image. That is, the missing half, each of the two nucleomolecules would therefore have one strand from the new material, this one, and the one strand will be from the old one, like the parental one. Look at here. This replication is called as semi. Semi means half conserved. Half conserved from the parents, but half is newly synthesized. Okay, what would be the third possibility, which is not accepted, but it's called as dispersive. Dispersive means dispersed like this, uneven distribution kind of a thing. So in dispersive replication, it was thought that it might be the way of replication. You can see the original strand, you can see the new strand, how it is scattered randomly with old bits and new bits thoroughly. So what will happen that in material in the two, parental will be the material of the parental strand will be distributed randomly between the two daughter molecules. So they thought that it might would happen, which is not acceptable basically, that the DNA broken down and the nucleotides replicated before and then being dispersed randomly to the new molecule. So what the new molecule will be containing? Look at here. The new molecule will be containing what? Both new and original material, but randomly distributed, dispersely distributed, which is obviously not the case. We have now done the whole experiment on this. So there was a very important experiment to basically justify the three types of replication. Okay, justify the three types of replication. What was the three types of replication? The dispersive, okay, the semi-conservative. I'm just writing in a short bullets. And number third is going to be full conservative. So we have to now check that which one is the true for the replication of DNA. So there was two scientists, Matthew Nelson and Stoll. You can also say that only for this we remember the Nelson and Stoll experiment for the proof of the semi-conservative replication. They used a very important bacteria that's called sometimes only E. coli. It's present already on your gut. It's very helpful for you. And this bacteria is going to help the alimentary canal digestion at the end. And where they grow basically what they did, the both scientists, they grew this bacterium in a food source which contained a very important, you can say, source of nitrogen that was called as ammonium chloride. NH4. They use that ammonium chloride for that. So they started growing these E. coli in that nitrogen source. So basically, the experiment was relied on the variation in the structure of the nitrogen atoms. As you know that nitrogen has 7 protons, but the number of neutrons can vary because they have different isotopes. So the nitrogen atoms have 7 neutrons. Their relative atomic mass can be 14. There are even another isotope which have 15. They have 8 neutrons. So they are basically forms of nitrogen. We use basically N14 and N15 for this experiment. They are basically called as the isotopes for nitrogen. Why we use? Because we wanted to give them the nitrogen source so that they can build up their protein structure in their body. So what they did, step one, basically they grew this E. coli in a nutrient broth, and nutrient broth first was containing heavier isotope, that was N15. So what did they? You know that bacteria grow by binary fission. That's a process of its division. And after every 20 to 30 minutes, what we get two daughter cells. After every 20 to 30 minutes. So what we did, we had many generations. After so many hours growing in this medium, what happened that the nitrogenous bases of the bacterial DNA, they were labeled with this heavier nitrogen, that was N15. Now they changed this broth. Now let's suppose first they were growing them in N15, the heavier one. Then they changed the broth of the same, E. coli, and they were switched to a medium which contained a bit lighter nitrogen isotope. And now they allowed them to grow for several generations. So first they kept on growing this, and then they switched this to this one. I hope the starting of the experiment is clear to you. Now what is the next, outcome they are expecting? Okay, so DNA which was made after that was switched to this N14 would have only the nitrogen which is available for the DNA synthesis. So what we did, these were the two, let's suppose medium, N15, and then grown into N14. Now we took this one DNA, we extracted it, we put it into a centrifuge machine. I hope you remember we did in the first chapter. This is called a centrifugation machine. It basically spun with RPM, revolution per minute. So it's going to have some values with G, gravitational value. We have. I hope you remember the heaviest organelles will basically obviously settle down the most, and the decant can be used again. And then we run that into the centrifugal machine, and then obviously we can basically use for the separation of the organelles. So what happened that we picked the DNA from now the E. coli which were grown in N14. We picked that DNA, extracted that DNA, and then we placed in a solution of cesium chloride, and then we spun it. We use that as a solvent so that the organelle or the DNA can be separated. Okay, now what happened? Because cesium atom is a bit heavy, it will sink toward the bottom of the tube during centrifugation, and density gradient will be established. Okay. Basically, we use this centrifuge machine on this principle that heavier will always, sink to the bottom first. Now the important thing starts from there. DNA obviously is a heavy molecule. It's present inside the nucleus. It will sink the gradient until they reach the level where their density equals to that of the cesium chloride. Okay, we are done now. Now, what happened? The DNA which was containing N15, they were a bit denser. They basically were, on the bottom first. And when they have a look on the N14, it was a bit lower level. But this is N15. So it was basically on the lower level. This is low. So basically the heavier DNA was closer to the bottom like this. And then it came to rest. But they also found out that there was some DNA which was present in the middle as well, like this. Let me just explain you this again. Imagine this is N15, the heavier one, and centrifuge machine have this principle that whenever it gets some heavier, and we decant off the material, and then we can reuse it into the next tube, and then we again spun it with RPM. So what happened that after a few spinning, we found out, let me color it with some other color, this one. It found out that this DNA was extracted and it was having N14, okay, which was obviously after the extraction of the